It's a good boy in a bad situation. But before I'm going to talk about that, I need a little bit of perspective on this talk I'm going to give here. And why I'm giving this talk is to try and give you a sense of the kind of research projects that Cure Alzheimer's is funding. And I'd start out by saying that this project would have had, or did have, a cinder's chance in snow of ever being done without Cure Alzheimer's funding giving us the money to do it. I launched this at NIH a few years ago and it took off like a lead balloon. And today they are giving me money to do it, but only because of that seed money that came from Cure Alzheimer's disease. And this is exactly the role that Cure Alzheimer's and foundations like it, I think, can play today, an absolutely essential role, because the public funding environment is so safety first, so risk adverse, that you just cannot get anything outside of the box to be funded. Let alone outside of the box, it's got to be in the middle of the box. Otherwise you don't have a chance. So this is a project I think that is innovative, it is out there. Some would say fringe, I prefer cutting edge. But in any case, the point here is that it has the potential to change the way that we look at Alzheimer's disease. Now, maybe that's an arrogance, but it's a healthy one, I think. So, Alzheimer's disease and A-beta. 27 years of Alzheimer's disease research on A-beta. That's a long time. Over 7,000 papers published on this particular protein. That's a lot of papers. And we know a lot about it now. Large body of data on the biology. It binds a whole bunch of different things. It's got complex trafficking pathways, modulated by in response to a whole bunch of environmental stresses, pro-inflammatory activities. But with all these complex physiochemical properties, I found 5 papers out of that 7,000 in the last 27 years that has even mentioned this issue. What is its normal function? Nobody has really even considered it. And I've gone around and talked to, the most prominent people in the field. Generally the answer is it's rubbish. Accidental product of catabolism. Doesn't do anything. Maybe it does something, who cares? And that's the thing. Why bother? After all, we know what we've got to do, right? We've got to stop A-beta. A-beta bad. A-beta bad. Well, in 1854, Dr. John Snow stopped a cholera outbreak in London. And he did that by removing the pump handle on Broad Street in London. He didn't know about cholera. He didn't determine that by intensely focusing on the disease in patients. And that, I think, is a great metaphor for this, because somewhere up there, upstream of A-beta, may be Alzheimer's disease pump handle. And if we don't go looking for it, we are never going to find it. So, I was thinking about this for a number of years. What could A-beta be doing? But my epiphany came when I discovered a little peptide called LL37. And the thing about LL37 is that it doesn't have the same structure of A-beta, but it has very, very similar biology. And this is a table. There's no way that you're ever going to look at all this and even understand or remember a fraction of it. But what I want to present it for, is to give you a sense of how many similarities there are. And also, I spent a long time on this table, so I like to present it as much as I can. Now, there are going to be a lot of tables, or a few anyway, tables and graphs in this talk. It's unavoidable. This talk is very much more focused on science, unlike Rudy's, which is a very broad talk. What I hope you're going to take away from it is a sense of what science is about, what the sort of projects we do and how we go about them, and how Cure Alzheimer's is helping that, is essential to that. So First thing is LL37 forms amyloid. LL37 is formed from a larger precursor that's chopped up. LL37 associates with ApoE proteins. It goes on and on and on and on.
You can say that again.
So, what the hell is it? LL37 is a member of a very ancient family of proteins, and I'm talking ancient here, back first multicellular, first glimmers of organised life. It's the foot soldier of the innate immune system, and it's an antibiotic. That's what it does. It's a member of a family called AMPs, antimicrobial peptides. And these AMPs are the chief effector molecules of innate immunity. What's innate immunity? It doesn't get a lot of press, or it hasn't. A few weeks ago, a couple of guys that did a lot of work on it got the Nobel Prize. And it's certainly rising in prominence rapidly. And the reason for that is, while it's not as sexy as its more famous adaptive immune system sibling, it's absolutely essential. It's not involved with antibodies. It doesn't involve cells secreting factors that modulate the whole animal. But what it does do is protect you day to day, moment to moment, against infection. And it's the mechanism that 99% of organisms on the planet use to protect themselves from infection. Hey, horseshoe crabs don't have antibodies. Earthworms don't have antibodies. Insects don't have antibodies. They have these things, and so do we. They are absolutely essential. And in fact, LL37, there's a rare mutation where you express it at low levels, and those poor people die in the first year of age from raging infections. And there's a bunch of others, similar stories. They're absolutely essential. They protect your lungs, your mouth, your skin. And without them, you're in big trouble. And there's a general perception in science also that there's a lot of them. There isn't. Less than 50 in us. In fact, there's only 37 in the big databases. By comparison, over 100 neuropeptides. So that's nice. A-beta and LL37. The central question, of course, well, does A-beta have this property? Well, obviously I'm here, so it does. And these were the first set of experiments that we did a couple of years ago. And they revolve around some pretty basic assays. We had a few false starts. I worked with a few microbiologists around the place. Nothing, didn't work. And then I was in the White Mountains with a family that my son was friends with from kindergarten. And I'm walking down the hill and I'm describing this to James, and he says, "You know what I do, right?" I said, "No, I don't know what you do, James. What do you do?" He was the director of microbiology at Beth Israel. That's when he said, "I can help you with this." So we went and did these experiments, and lo and behold, indeed, A-beta is an antimicrobial. And this is a kind of example of a simple assay. You put bugs in a plate. Where the bugs grow, they sink to the bottom, they form a little pellet. You can see it, hold it up to the light. Then you put your peptides, your proteins, LL37, A-beta, whatever, increasing dilutions. Where they don't grow, you're getting activity. Where they do grow, you're not getting activity. And the point at which that switches over is called the minimal inhibitory concentration. The smaller the MIC, the more potent it is, the less you need to kill the bugs. And if you look at a battery of bugs, a library of pathogens, and you line them up, LL37 is an archetypal AMP. It's been the subject of numerous clinical trials trying to bring it to use as a clinical agent, hundreds of millions of dollars. A-beta stands shoulder to shoulder to it on its potency against a bunch of these organisms. And notably, many of these organisms that A-beta affects, they're the ones that give you meningitis, exactly what you'd expect, if that's its real function, to function as an AMP.
Let me interrupt you for 1 second here, because you just skipped over an important point. First of all, you should explain on the chart, the left-hand side are all bacteria, and a peptide is a portion of a protein.
That's right.
You're showing that two kinds of A beta have antibacterial activity that are equal to the other one. But the point that what I was skipping over was that the big excitement when you wrote your book was about a vaccine that was made by an Irish company that was given to people to cure A beta, to eliminate A beta from the body. So volunteers who were given this vaccine, seemed like an amazingly simple concept. It is a piece of a protein, a peptide. You want to get it out of the body, so you give a vaccine. It did, it worked, it got it out of the body, and what did the people get? They got meningitis, brain inflammation.
That's exactly right.
So,
yes, increased rates. So there's quite a bit of that out there that people they walk up to the door and then they go sideways and miss the point. And I'll get to a couple of experiments like that later on. So yes, what we're looking at here, smaller numbers, better. And A beta, there's a number of different forms of A beta. They're called 40 and 42. But essentially, what that's telling you is that for a lot of these bugs, LL37 and A beta have equivalent activity, or indeed A beta has better, particularly the top one, which is actually yeast. Now, probably 20% of inmates in nursing homes have chronic albicans infections in their nasal passages. Interesting point. So, the other thing we did, that's nice. Okay, so A beta kills bugs in a test tube. That's good evidence, but not the only evidence you'd need to make the case. In fact, it's a long way short. But another way we could look at this is to take Alzheimer's disease brain and normal brain and actually add it to cultures of bacteria and see if it kills them. It seems like a pretty simple experiment. It kind of is. And the results are quite startling. If you take bits of brain that are heavy in A beta versus bits of brain that have very low A beta, you see a distinct difference between Alzheimer's disease and normal patients. Alzheimer's disease brain is an antibiotic. It kills bugs better than normal brain. And you can prove through various mechanisms of immunodepletion, various other ways, you can pull out the A beta from this brain. If you do that, that effect disappears. It's A beta mediated. This activity is due to the A beta in the Alzheimer's disease brain. That was pretty strong evidence. But of course, in the world of science, what people really want to see is this working in living animals. So we published this about a year ago now. And I have to say, even if this whole idea fizzles, I still think this project has earned its stripes. That is because that paper has had 15,000 views now. And it has stimulated the interest in that basic idea. What is the function of A beta? What is upstream? Is there an Alzheimer's disease pump handle just sitting up there? And maybe this is not it, but now that there's a lot of interest up there, maybe somebody else will find it. Frankly, I think this is true. And the reason I think that is because of the last year's experiments. So what I'm going to show you now is actually brand new data. And it involves looking in vivo, that is, more in living systems rather than test tubes. And the first model we used, very simple model. You take cells, human cells, you grow them on the bottom of a plate, and they form a nice layer, very convenient. And then you just pop in some bugs, yeast. And then you watch what happens. There's a simple diagram, a picture here. This is a culture well along the way. A lot of cells are starting to die. The yeast's taking over, essentially. If you look close up, what is happening here is that the yeast is glomming onto the cell, sticking its hyphae into the cell, literally stabbing it. And the cell eventually bleeds to death from this kind of damage. And you can track what's happening here. And if you look at markers for cell damage, and you have cells that express A beta in there versus cells that don't express A beta, they're what's called transgenic cells. They've been made with extra copies of the human A beta gene. They pump out A beta. You see a startling difference, and that is cells that express A beta show less damage. That's after about 10 hours. But the real striking thing is what happens at about 40 hours when the cells start to die. And that is the A-beta expressing cells don't die. They keep going. Now, that may not look like a startling difference, but think of that as brain cells, and you start to get to a sense of how important that is. So that's nice, cells, but once again, that's reasonably convincing, but not enough. So we've been working with another model, a very popular model among scientists, fruit fly, Drosophila, and that's him there. Now, once again, you can make what's called transgenic fruit fly. You take a human gene, you put it in it, and it expresses it. And we have a model like that. It only expresses A-beta in the eye. So we took that fruit fly and we jabbed it with a needle, inoculated it with yeast. This is an old model, been around for 25 years. Most people use it to look at the virulence of the yeast. We switched it around to look at the ability of the fly to resist the yeast. So we have normal flies and we have A-beta expressing flies. So if you look at the normal flies, you see a difference here. The red line is what happens when you take a normal fly and jab it with yeast and infect it. The blue line's what happens when you just jab it and it has nothing on the end of it. There's a clear difference there. It doesn't look big, but we're talking thousands of flies here. This is a highly significant difference. A lot of counting. A lot of counting. So every day you've got to look at the 1 dead, 2 dead.
We have students.
Yeah, we just you've got students. Anyway, so what happens when you look at the A-beta flies? Bingo, no difference. They're protected. They're protected from that infection. Once again, exactly what you'd expect if the function of A-beta is as an antimicrobial peptide. Now, that's one fly model. None of these A-beta models, animal models, are really perfect. They all have their problems. So we've been looking at a second fly model. This one has a more global expression of A-beta. It's not just in the eye, it's in the entire central nervous system, which is good and not good. It makes them pretty sick. Yeah, it's not a good thing to express in your nervous system all the time. So they're very fragile. They're not very healthy flies. But if you infect them with yeast, even though they're more fragile, they survive much better than the wild type. Really surprising result. Once again, consistent with the idea that A-beta is an antibiotic. That's its function. We have a third model, which you can switch A-beta on and off. We've just started working with it. That one we hope will overcome a lot of these issues. But when you talk to scientists, of course, what they really want to see is a mammal model. They want to see it done in mice. We are doing that. That's this coming year's project. But interestingly, it actually has been done, but by accident. About 6 years ago, there was a paper on this. They weren't looking at this. They were looking at something quite separate. But I will say the advantages of a mouse model is that it will really be the clincher. It's the acid test. You can have two different types. Firstly, you can have a mouse that doesn't express A-beta, including its own native rodent A-beta, because mice make A-beta as well. It's a very old peptide, very old protein. If it doesn't express it, you'd expect it to be vulnerable to infections. You can also have ones that overexpress it, that they have a human gene shoved into them. You'd expect those to be protective. Well, about 6 years ago, a model was created of mice that knocked out BASE. BASE is an enzyme, and it's what's needed to create A-beta. It chops up the larger parent protein of A-beta. When there were two forms of it. So when they chopped out the first one, BASE 1, they got these mice from another lab and they were experimenting with them. They tried to grow a colony. 40% of those pups died in the first 2 weeks. And when you knocked out both, 60% died. They couldn't work out why.
Rob, We're going to have to wrap this up a bit so we can get some of these questions.
Okay.
But we'll leave that up for a second. The fourth to the last author is named Alzheimer on that paper. Did you notice that?
No.
Who is that person? Anyway, so just thought I'd point that out.
All right, so I'm going to move along a little bit. Okay, so does that mean that AD is caused by infection? I don't know, maybe. There are really three possibilities. Firstly is, yeah, A-beta, Alzheimer's disease is caused by persistent CNS pathogens. Certainly there's a bunch of guys who have been out there screaming that for a while, which we've all ignored, but maybe they've got a point. The other thing is maybe it's a transient infection. Maybe it's something that happens early on, kickstart the whole process, and it won't switch off. And the third possibility is more in line with the current thinking, that is it's a dysregulation for something happens, not related to its function, but it causes A-beta to be generated in large amounts because the whole system is out of whack. So that's a possibility. I don't know which one it is. But if I can show that A-beta is indeed an antimicrobial peptide, part of the innate immune system, that opens up a whole bunch of drug targets, all the innate immune system, and there are a lot there. And then the last thing is, I'd like to mention the implications beyond Alzheimer's disease. So there's a lot of amyloid diseases out there. We mentioned diabetes, type 2 diabetes, 27 million Americans. It's an amyloid disease caused by a protein called amylin, very similar to A-beta in a lot of ways. Obvious question, is amylin also an AMP? So through Cure Alzheimer's Disease, we got funding from the Helmsley Foundation to look at this. And we've been looking at it in the last year. And the answer is, you betcha, a very potent one. And it goes after the bugs that are normally found in pancreatic infections. There are 30 amyloid diseases out there, at least. I don't know how many involve AMPs, but clearly this paradigm could be very important for all of them. So lastly, I'd like to thank a few people. Stephanie Sosha is actually the Ph.D. student, did most of this work. She got the Boston University School of Medicine Dean's Award actually for this work. She got awarded a Ph.D. this year. And of course, Rudy, who a lot of this stuff we thrashed out on his deck over a very nice bottle of plonk over a couple of summers. And on that note, I'll thank you.
Tell us, you talked about bacterial infection, but inflammation doesn't just occur in response to bacterial infection. It occurs in response to injury as well. So tell us, there is, and Rudy used the metaphors from both football, Patriots Stadium, and the Gillette Field, I guess, and soccer games. And we're thinking a lot these days about former NFL players, retired NFL players, getting Alzheimer's disease. So what does this tell us about this connection between head injury and Alzheimer's? And there's also another study coming out in veterans showing that those who studied head injuries are more susceptible to Alzheimer's.
Yeah, well, the neat thing about this, of course, is that it does incorporate many of these ideas, infections, blows. The innate immune system is not adaptive. It basically has the same response to whatever insult you throw at it. There's a couple of things I didn't mention. Why brain? Well, brain is an immunoprivileged organ. Antibodies don't get in there. AMPs are the only game in town. Yeah. So, whether you've got an infection, whether you get bonked on the head, whether you have a stroke, the innate immune system basically does the same thing pretty much every time. And that could be ramp up AMPs and give you, in the long run, if it can't ramp them back down, Alzheimer's disease.
Well, we know that amyloid beta protein does go up with all of these, head injury, neurotoxins, infection, stroke. His data would suggest that it's deliberate as an AMP in the innate immune system trying to protect the brain.
Well,
now,
so if that's the case, that sounds troubling to me, because if we don't want to get completely rid of A-beta. We want to have just the right amount.
Exactly.
How on earth are we going to do that?
We do it with cholesterol.
Yeah.
So it's, okay.
And
That's the model.
And the other thing is that the implication beyond that is that it may be possible to go way upstream, that pump handle, and tackle the events that initiate it. Now, it may turn out that they are infections, that it is infections that get it going. If you could treat that or target that or switch it off,
Vaccinate against it.
or switch it off at that point, and if you can lower the whole system down, it may just quieten down of its own accord. It's this constant belief that it's got an infection raging.
We have a lot of questions from the audience about something you talked about earlier, Rudy, and this idea of exercise and the connection to Alzheimer's as well. You also mentioned intellectual, both of you, David, mentioned intellectual activities. I think, first of all, based on what you know now, which is more important in terms of what you can do to reduce your risk of Alzheimer's?
Well, based on what we know from animal models, physical exercise.
Physical exercise.
Because physical exercise triggers the breakdown of amyloid. So over decades, see, the amyloid that gets made in the brain, some of it just gets so clumped up it can't get out. But exercise seems to trigger its breakdown.
So the same health message that Americans have heard for so long now about avoid metabolic syndrome by exercising, keep maintaining a normal weight, all these things are critical. And we haven't done a very good job as a society of bringing those things under control. The number of diabetics is going up even faster right now than the number of Alzheimer's cases. And as a result, do you think fear of Alzheimer's, or is there some way that we can put a campaign to get people to understand this more, that if you don't get yourself into shape, if you don't have any physical exercise, that you're putting yourself at risk for this horrible disease that's a burden to your entire family?
Yeah, I think a message that the 4 big diseases that threaten healthy aging are Alzheimer's, heart disease, cancer, diabetes, and include stroke there with cardiovascular. All of them can be prevented, or at least lower the risk with exercise. So if you look at what really threatens healthy aging or health span, knowing that lifespan is now 80, exercising and just keep moving would seem to be a great way to try to prevent.
So yeah, isn't it true that if you could put exercise in a bottle and sell it as a pill?
Yeah.
No, seriously.
We thought about that.
No, but people want to do that. But seriously, if it were, it would do far more to reduce, right now, to reduce the risk of Alzheimer's, stroke, heart disease, and anything else that you could do. If you could get everybody in the country to do, how much exercise is the right amount of exercise?
So
You're not a physician.
There are 2 ways to do it. If you think about exercise, you're getting more blood flow to the brain. So there are some folks who think it's just getting more cerebral blood flow that's helpful. And there are others who believe that it is based on the fact that with exercise, you actually make new nerve cells. So something called neurogenesis, making new nerve cells in the hippocampus. So the very region that's affected in Alzheimer's with short-term memory, hippocampus, is where you make new nerve cells by neurogenesis in response to exercise. That was shown ages ago by Rusty Gage and has been replicated over and over. We have a fellow in our lab who's funded by Cure Alzheimer's Fund looking at how neurogenesis in response to exercise leads to the decrease in amyloid, and it does. So it may be a combination, blood flow, new nerve cells being made, and then you get rid of the amyloid. So we're looking into the molecular mechanisms of that now.
There's a lot of curiosity in the audience about when you're testing these drugs in human beings, not in mouse models, because a lot of things have been cured in mice, and not just in Alzheimer's, unfortunately, that don't end up being cured in human beings. But when you're curing them when you're testing the drugs in human beings, what's the process? How do you know if something's working?
You have to improve cognition and good old-fashioned psychological psychiatric testing. So you can look at amyloid in real time by imaging. You can look at what are called biomarkers in the cerebrospinal fluid to indicate you're doing the right thing. But in the end, the FDA, to approve a drug, will want to see evidence that the person is improving at the cognitive level.
So taking those simple tests where you count backwards from 7 and put together triangles and all that stuff,
That's still the gold standard.
That's still the gold standard.
Well, yeah, the FDA will not just accept data on imaging or biomarkers that reflect what's going on. You have to show cognitive improvement. And the fact is, for the drugs we now have, if you use a ski metaphor, that these drugs may take you off the blue slope and put you on the green slope. Okay.
Temporarily.
But is
temporarily, yeah. Until you hit one of those blue slopes, you didn't know.
I mean, it's important to point out, sticking with your cholesterol metaphor, that there was a drug approved by the FDA on the basis of lowering cholesterol that turned out to cause more heart disease than it prevented. So that's why this danger about using so-called biomarkers instead of progression of disease comes in. Yeah. Because it was great. It looked, oh, this is terrific. It raised HDL, the good cholesterol. It lowered LDL, the bad cholesterol, and it killed people from heart disease.
Great example.
So it wasn't such a good drug, and it's been taken off the market.
You can use biomarkers and imaging to know that you're pre-symptomatically starting to get in trouble, so you can start drugs preventatively in time before the disease strikes. But in terms of showing that a drug works, you really have to show that it works on what you want, which in this case is improving cognition.
And on that subject, and any of you please jump in here, there are questions because of this push to intervene earlier in the disease process. There's a lot of work being done on imaging people and taking spinal taps from people and blood samples as well, or looking in their eyes and that sort of thing, to try to find if they're at risk for Alzheimer's before they have any symptoms. What's your advice to people who are approached about that kind of thing, unless they want it? Should people want that more actively, or unless they have a strong family history, or do you have enough volunteers for subjects for the experiments you need?
Recruiting for trials has been, not a big problem, but recruiting the right patients. I mean, you mentioned early on you have to recruit patients at an early enough stage to treat them, but you also have to make sure they have Alzheimer's disease and not some other form of dementia. So the biomarkers and imaging help there. But people shouldn't think that we now have tests that can surely tell you you have Alzheimer's before you have symptoms. It's still a work in progress. The press does, no offense, the press does does overhype it a little bit. Not you, Bob. No, your blog is always actually right on time.
And none of your colleagues has ever exaggerated his work.
No, none of us.
For the purpose of getting attention. What has to be the final question is about genetic testing. You have discovered, and there are and others have discovered this enormous number of genes. What should people do about genetic testing for Alzheimer's right now? Where does that stand?
Well, the fact is we don't have a genetic test for garden variety Alzheimer's. We don't have it yet. There are direct-to-consumer companies that will test your DNA and tell you your ApoE results. But ApoE is not sufficient to predict the disease. ApoE is working together with lifestyle and other genes. So with the exception of these 3 genes where you have 200 or so mutations, as you said, in about 300 known families that cause the disease directly, you can do genetic testing there. And it's done. But for 98, 99% of Alzheimer's, one of the whole points of the Alzheimer's Genome Project is to find the rest of the genes so we can have 100, maybe more of these on a chip and do a more responsible test. And I say this because some of the genes, about half of the genes we find contain variations that increase risk. The other half contain variations that protect you. So you need to do them all. We only think about testing for risk genes. You also have to test for the genes that protect you, and in the end come up with a very sophisticated calculation or algorithm for how your protective gene variants and your risk gene variants combine to determine your lifetime risk. That's not easy. I know that direct-to-consumer DNA companies make it sound easy. They're just going to do your ApoE, but it's not sufficient. So the fact is we don't have a real reliable genetic test yet. That's still in the works. And my guess is for how long that's going to take, it's probably going to dovetail with when we're going to have drugs that will efficiently empower people with protection and prevention against the disease. And that's probably going to, again, probably take 5 or 10 years. Excuse that term.
Well, thank you very much. That was thank you all for your support for the Cure Alzheimer's group, and thank you for coming, and we appreciate it very much. Thanks to the panel.